Silence Therapeutics is a world-leading genetic medicine company. So I was actually wondering over the weekend whether that's actually true, and how many CEOs and companies stand up at events like this and say the world-leading, world-class. And I guess what made me feel better is I thought, if the audience went of their own volition and kind of looked it up and tried to measure this, I think the answer would be yes. And so I'd start off with some of the questions you could ask. Do we operate in one of the most important and biggest sectors in medicine? Do we have critical patents protecting our IP and technology? Is there a very high barrier to entry, massive barrier to entry? Do we have experience of our drugs and agents in humans rather than just in animals? Are we a preclinical company, or will we further? Do people want to collaborate with us, I guess, is another important question. And how many of these us are there? And I think if you do that investigation, and I think maybe if you have time you should, you will unequivocally find Silence Therapeutics. The answer is yes. Okay, so Silence Therapeutics is a world-leading genetic medicine company by any reasonable measurement of technology, IP, and in vivo data. But I think best of all at this point is technology is now coupled with and lends itself to one of the most simple and effective business plans for me in any business sector, let alone just biotechnology. I believe we can make drug development faster, specific, and significantly less risky. And I'm quite aware that's quite a bold claim to stand up here and say in a sector and industry which is inherently volatile and full of failure rates, in particular in the UK. Okay, so I'm going to go through the technology behind the preclinical R&D engine, as we call it, or our genetic toolkit, and the details of the business plan which we think is so compelling. And actually, more importantly, the business plan which is being implemented today is in action today. And I'm going to go through Silence Therapeutics of, let's say, December 2013 to the Silence Therapeutics of today and the changes that we've made. So to really appreciate the full, if you like, power and flexibility of our technology, if you just bear with me, I think we need some basic biology lessons. And no better starting place than the central dogma of biology, which states that DNA is transcribed first to mRNA and translated into protein. And more specifically, think of DNA as an encyclopedia of genes. There are roughly 22,000 genes in the human genome, of which approximately 2% are what are called coding genes, the rest of the 98% performing regulatory or other functions. So of this 2%, each gene has a different code to make, or a different instruction to make a different protein. So if you want to be slightly more specific here, in this example, one gene is first transcribed onto this temporary system called the messenger RNA. It's got its own code depicted by the colors here. The cell's natural code-reading machinery, if you want to say it, translates this code into a protein. And so the process of cell life goes through and through repeatedly like this. And at this point, a good pausing moment is the target of our drug is messenger RNA. And that's something which will become more and more apparent as we go on. So going into a cellular view of the central dogma, here we have a cartoon depiction of a cell. Again, DNA within a nucleus, it's over here. The encyclopedia, one gene being transcribed, specific code to make a protein. I'll come to this gauge in a minute. If we go to a disease state, this is just one example of a disease state. One gene is not being, there's multiple copies of this messenger RNA, therefore there's multiple copies of protein being made. And you can think of this as an overactivity or an overexpression. If you want a real-life example of this, think about an overactive loop, such as cancer, leading to tumor growth and progression. So kind of think overactivity and proliferation. And it's at this point that our therapeutic activity begins from outside the cell by the introduction of what's called a short interfering RNA, or an siRNA. So you can think of a short interfering RNA, as the name suggests, short in length, interfering, as in interferes with mRNA. As an anticode, or let's say the perfect opposite sequence to the mRNA is trying to target. So think opposites attract negatives and positives. And if we go into slightly more detail of the mode of action of how this siRNA actually works once it gets cellular uptake. So we start with phase one, where the siRNA is within a cell, it finds the messenger RNA, and the codes start attracting each other, again, opposites and negatives and positives. At this point, there's a binding process. Again, think the opposite attraction. And now this binding process triggers something very important discovered by Fire and Mello in 2005, which is the RNA interference pathway. And the binding triggers what's called gene silencing. So the cell's natural silencing machinery spots this binding and starts to literally cut up or degrade this messenger RNA. This cutting up process stops the messenger RNA being read, protein production is reduced or even stopped entirely. Whoops. So if you want to use this very simple gauge, in the healthy state you can see the gauge is on green, reasonable, normal activity. Over here it's on red, too many, think about overactivity, overexpression, etc., etc. And once the gene silencing effect comes into place, this gauge should go back to normal. You should also remember that we can, in effect, synthesize externally an siRNA or an anticode to every gene in the human genome. So since the Human Genome Project, in public databases, every gene and every code is basically well known. So the synthesis of the anticode is particularly simple. So a more practical example of our therapeutic mode of action, let's go to an indication or our clinical program, AT2027, metastatic cancer. We have an internal hypothesis which starts saying, if we silence a gene called PKN3, we can improve the metastatic rate, slow it down. This is not just internal work, but also validated with external work as well. So the target mRNA is PKN3. Here's a depiction of the PKN3 gene as seen in the databases. We synthesize the opposite code. You can see the different colors. That's how we've done it here, PKN3 siRNA. The natural cell machinery takes over, and we have what's called a silencing effect. So the silencing effect, this bar chart, is simply a measurement of messenger RNA, the number of messenger RNAs or protein production. So a simple point is, let's say we start here in a control setting, which is the gene doing what it's doing. When an siRNA is introduced, you can see this significant reduction. Look at another one of our projects, pulmonary arterial hypertension. Hypothesis, again, well known in literature and with some in-house experiment work. Silencing endothelin-1, ET1, will improve function. Same principle again, ET1, well-known code, ET1 siRNA, synthesized anticode, silencing effect. And just slightly over-elaborating the point, acute liver failure, again, silencing the FAST gene. FAST gene is pretty much public information. FAST siRNA, synthesized, silencing effect. So actually it transpires that one of the limitations, and ironically one of the almost opportunities in our technology, is that simply introducing what's called a naked RNA molecule, let's say an siRNA, into a human system or an in vivo system has certain, let's say, issues surrounding it. And as such, you need specialist delivery systems, which are depicted here in this graphic. These are our delivery systems, the broadest delivery systems in RNA interference, where our competitors are heavily active in the liver. We have delivery systems to various different tissues and structures. And obviously, these are really, in some ways, Silence Therapeutics as technology really is in this slide. This is highly proprietorial, fully IP protected by patents. And this is really what separates the kind of up-and-coming companies from the companies in what I call this exclusive club. Okay, so now we're getting a good picture of the components of our drug. It's a two-component system. It's a delivery system, and in this case, an siRNA. So again, we've divided it up into two. We have a delivery and an efficacy section. So this is what's called a biodistribution chart. Maybe this is a good starting point. We are measuring where the siRNA is ending up in vivo. And here you can see by this bar chart that the majority of the drug using our DAC system ends in the lung. In our hepaplex and macplex system, the siRNAs are ending up in the liver. So clearly lung and liver indications are important to us, and we want specificity there. And if you want a more broader delivery in disease settings, for example, like metastatic cancer, where you need siRNA and silencing effect, in a broader setting, you might use Atiplex. And here is a familiar bar chart and the silencing, or otherwise known as knockdown effect. So we're basically taking a control, we apply an siRNA, and you can see percentage reductions in different tissues. Again, we can target any gene in any of these tissues using these delivery systems. Okay, so today we made a very important technology breakthrough announcement in mRNA replacement. And to really understand what's going on here, I think we just have to revert again to the central dogma. But a good starting point for you to think about is mRNA replacement is the complete opposite of our siRNA therapies. So if we go to the central dogma here, DNA transcribed to mRNA to protein, you can see the mRNA here is completely missing. So a reason for that might be the gene is literally missing. Think genetic deficiency diseases, for example, cystic fibrosis, muscular dystrophy. So again, we go to this two-tier system again. We have a delivery system. Again, we're showing where the mRNA we're putting into a cell is ending up. In the DAC system, we have lung. In the liver system, hepaplex, we have it here. And so what's happening here is when we introduce just a delivery system with just literally sugar, protein production is missing completely. There's literally nothing being made here because there's no gene to make it. There's nothing to read. However, when we introduce the mRNA, so think again, the mRNA is a code, it's public information. We put it into the delivery system. With our DAC system, it ends up in the lung. In the lung, it ends up in the cellular uptake, it ends up in the cell, and the cell's code reading machinery spots this messenger RNA, which was previously missing, and starts to making functional amounts of protein. And you can see we have the same principle in our liver delivery system. So obviously, this, if you like, upregulation, so turning things on and off, has opened an enormous new set of potential therapeutic areas which were completely closed to us: genetic deficiencies, vaccines, infectious diseases, all that kind of stuff, cellular reprogramming. And I think, even though it's a headline term, we can say something very important in Silence today, that we are capable now of modulating gene expression both up and down in vivo. Up in order to replace genes with messenger RNA, and down using a short interfering RNA. So the net result of all this is a familiar slide for some of you, is our genetic toolkit or our preclinical R&D engine. So we take different payloads, and I've talked about short interfering RNAs or messenger RNAs. Won't be part of our presentation now, but microRNAs is also an RNA payload we're working in. We mix and match it with our existing delivery systems. Again, remember the broadest delivery systems in RNA therapeutics. So if we need a lung-specific disease which requires silencing or taking overactivity down, we'll use DAC and an siRNA. If we're looking at a liver condition where, for example, you need to basically put something back, we'll use hepaplex and messenger RNA. And we take our scientific expertise and the testing facilities internally, and you can see some of the, quite famous collaborators that want to use our technologies, maybe because they don't have access to delivery systems and they have certain models they want to work with. And this is our sort of engine and preclinical R&D engine, and we think this is a very powerful piece of technology here. Okay, so what is our drug development strategy and the business plan I've talked about so much, and what progress have we made, I guess, since the last update? I think it was in November. So I'm going to start with some pretty damning statistics of biotech and drug development as it is, with three graphs really. Top left, we have soaring costs adjusted for inflation. 1970s, roughly $180 million to make a drug. You can see the figure is still to $2.5 billion. The approval rates are roughly running at 20% at the moment for any drug that enters the clinical trial system. But I think for me, the most shocking number really is fewer than 40 new molecular entities, or basically new drugs, are approved every year. So when you consider the multi-billions of dollars of R&D which are being done globally in the commercial world, let alone the academic world, by any definition, this is a tough business to be in, and this is a tough business to invest in. And frankly, I'd go as far as saying maybe biotech in some cases is uninvestable in. So clearly, drug development is in need of a new business model, highly target specific, shorter timelines, and multiple shots at goal. So rather than having just one candidate or one program that you get behind and you have this kind of zero to hero situation. And I'm going to make my second bold claim, which I think we can turn these numbers on its head. Okay, so let's start on this journey of trying to back up this claim with some numbers and reality. Here we have a comparison of small molecule drugs against an RNA therapeutic. And rather perversely, let's start backwards. So you can see that at the clinical trial system between the two, at the FDA review and approval, and even at the preclinical stage, there is not much time saving or even necessary cost saving between the two. I mean, there are ways now you can speed the system up: orphan designation, breakthrough status. If you're doing particularly well with a compound, you might get a pass from the regulator and kind of speed through. But where you begin to see a huge difference is at the preclinic and actually primarily in the drug component of actually making a drug. So in a small molecule, you're looking roughly 4 to 5 years and the cost of roughly into the millions of dollars. Whereas for an siRNA or an mRNA therapeutic, once we've found a target, let's call it endothelin-1, we can screen, find the best siRNA, and put that into a delivery formulation in the cost of, as you'll see later, thousands in 3 months. And I actually think this slide is a starting point for why the failure rates are so high. You think if you're in this industry, you have spent 4 to 5 years just making the drug before you've even ever seen any efficacy of it, to see even if it works in any kind of model, and you spend millions of dollars, you're highly incentivized to keep the show on the road. Over here, it's the complete reverse. You're highly incentivized to go with your best program. So this time last year, we tried to do a sort of root and branch review of what our company kind of looked like, what the preclinical R&D engine looked like under the bonnet, and what we could actually do to sort of streamline the process of drug development. And this is Silence in December 2013. And it's worth spending a bit of time, just a little bit of time actually understanding what's going on here. So there are quite a few moving parts, right? So the first assumption is, call this the preclinical R&D engine, the workings of it. And let's say the translational team, which we have in effect, has worked up an idea and says, right, we think that endothelin-1 and pulmonary hypertension is something we should be investigating. So we think as this idea, if you like, enters over here at Q1 in any given time, we can, within 18 months or 2 years, be in a phase 1 in an IND setting for a total cost of 1.7 million. And we've broken down the processes into really 3 or 4 very small, very easy to understand moving parts. Right? So as we discussed before, the siRNA and the lead identification and putting it into a delivery system is particularly simple, takes 3 months. Remember the small molecule example, 4 to 5 years. Once we've got our best siRNA against the target, ET1, we basically go into what's called an efficacy in animal models. We try and find the best representation of pulmonary hypertension in 1, probably 2 animal models, in rodent models, and show that we can beat either the gold standard or have a material effect on disease. And if we do that, then it progresses through the engine into standard non-human primate studies. Just notice how the costs start to go up at this point. We start to make clinical grade material, and our clinical team starts to apply for a submission. So in terms of costs, I mean, the first striking figure, as I said to you, this is in the thousands. That is a material cost saving and fast throughput industry relative to other drug development. But I guess for me, the best number is actually this interim go, no-go for 350,000. So at this point here, we've synthesized a drug, we've been in 2 models, and in the space of 1 year, we can have a high confidence level because of the specificity. It goes back to the binding process. 1 siRNA targets 1 messenger RNA. We can, with quite a high degree of confidence, either go ahead with a project or kill a project at this point. So I mentioned the translation team, and this is how we're set up at the moment internally. We have a technology department, which is basically based in Berlin. The technology department is working on delivery systems, optimizing delivery systems, new delivery systems, evaluating delivery systems, mixing and matching with payloads, and showing proof of concept that you're targeting cell types and you're achieving either knockdown and silencing or replacement. And what the translation team in effect does is take the technology and start adapting it with market information to where we can apply it, which diseases we can apply it to. So we provide an algorithmic process which we come to, and that then is fed into the clinical team. So the net result of that is in Silence in December 2014 is, as you can see, a materially different company. And we're starting to see the beginnings of the implementation of a multiple shots at goal business plan. And here really we're following, this is our siRNA graphic, we're following a very simple formulaic or algorithmic approach. We identify an indication, let's say again endothelin-1 and pulmonary hypertension, identify the target gene, we ask the technology people, can we deliver our siRNA or our agent to that cell type? If it's passing these points, we go and synthesize the siRNA for the cost that I said, £30,000. Does it pass the go, no go? And we've tried to apply this formula to the whole company. And you can see that the translational team at various different stages is starting to materially change the kind of optionality of Silence Therapeutics. And we'll come to a slide on delivery, but subject to delivery limitations and number of targets, there's no reason why this slide can't be populated with tens, hundreds, if necessary, of indications. Now, one caveat I will put, I would say that these are what we call internally as projects and not IND-enabled projects. I think once they cross this line, we start to get into the IND scenario. But you can see, we're beginning to see a different type of biotech at work. It's diversified as an investment proposition. You're not looking at these kind of single shots at goal. You're not backing just one candidate and hoping that it basically, frankly, stumbles through. You're incentivized as a management team to actually kill projects rather than go ahead with them, which makes a material difference to the outcome. In effect, you go with your best ideas. And of course, coming up, as I said, delivery system plus messenger RNA, our technology breakthrough, all feed into exactly the same model. Almost a factory line. We go into the same thing. Instead of an siRNA, we formulate a modified messenger RNA. MicroRNAs, we want to basically develop exactly the same strategy. Okay, so I hope I've gone some way to convincing you that my bold claim earlier that we can in some ways reinvent this industry is potentially true. So we have a few bullet points here. I think the simplicity, I hope, has come through. The simplicity, 30,000 pounds to synthesize a drug. The specificity goes to the mode of action. One siRNA binds to one gene, and a messenger RNA, if it has cellular uptake, the machinery reads the code and makes it. Translates to lower risk. The lower costs that you've seen because of the synthesis costs and the simplicity angle. The reduced timelines come into play. And then finally, the modular technology, this mixing and matching of literally payloads with delivery system to different tissues and different cell types. Okay, so I promised a few slides on delivery. So the opportunity in delivery is that it actually provides a very high barrier to entry to any competition. So if you do your investigation, and I think I've even given some of you charts on who is in this club and who is one of us, I think one of the great attractions of our industry is it's very hard to break into this club. We've spent 15 years really optimizing and tweaking these delivery systems with in-depth experience and maybe even frustrations with RNA molecules and how they react within them. So the opportunity really is in the fact that we're kind of blocking people from getting into it. The limitation is that at the moment in RNA interference, for all the progress is made, there are only 2 tissues and cell types that anyone in the world can deliver payloads to. The liver, which our competitors are highly active in, and the vascular endothelium, which is our main operating area. So even this limitation, for me, is actually a huge opportunity, because these are the structures and organs that are all to play for, if you like, are game-changing delivery locations. Think of, in terms of the brain, dementia, Alzheimer's, a loss of function, probably quite good for messenger RNA replacement therapy. Heart, cancer cells, and you can see that each one of these organs and tissues has genes that need to be either inhibited and silenced or at least replaced and made active again. So we have our very simple approach to our delivery technology and our business plan. Internally, we are constantly optimizing and further profiling our current delivery systems to see where the pressure points are. We're adapting our delivery systems to new RNA payloads and hoping to see improvements in those. But importantly, we're obviously keeping an eye on the future externally. We're constantly evaluating delivery systems, either to the cell types that I've mentioned that we haven't got access to, but actually, kind of more importantly, we have a very reliable benchmark, in-house benchmark of our own systems to test against. Any systems that come in and start to score against it, do better in it, we know they become in-licensing, out-licensing opportunities. At the moment, I think we've got 10 external systems currently being assessed in-house. And obviously, we're investigating alternative, what's called non-liposome-based platforms, which is our main technology at the moment. I wanted to give just a quick slide on the type of work that can go on in a short space of time. So I think since our last update, the first chart I have is on our mRNA side, and this is a chart of time progression with protein production. So you see over here, we're putting an mRNA back into a cell, we're starting to optimize. Around May 2014, we were beginning to see flickers of life in protein production. And you can see that in the space of 6 months, through optimization and frankly hard work and graft, we're really starting to see some dramatic changes, opening up completely new therapeutic areas. In terms of new horizons and new frontiers, macrophages are key immune cells. We're beginning to see excellent knockdown. You can see the bar charts that I've talked about, knockdown at certain days. And so immune cells are becoming open to us. It's early days here. But obviously, going back to the limitation, we're trying to move from the limitation of the endothelium and the liver into new areas. Okay, so in terms of our clinical programs, our clinical trials aim to reveal both hints of efficacy and indication of a target knockdown in man, a silencing effect. But I think it's important just to have a little pause here at this point and really appreciate that Silence Therapeutics is not just some technology or preclinical or research organization. Our core siRNA molecule, which is used to silence gene, and our Atuplex delivery system has been in man, has been incredibly safe. Our core molecule, the siRNA, has been tested in more patients than any other company in the world. 400 patients have had no serious adverse reactions to it. And I think that's a very important valuation marker for this company again, because in nice colloquial terms, it separates the men from the boys, if you like. But nevertheless, we'd like to give you some updates on our clinical program. We're pleased to say we completed recruitment in July 2014. We have, again, very encouraging safety profile for long-term treatment, and we hope to give you an update on the findings in Q2 2015. As you know, we're starting a head and neck trial, probably starting in Q1 2015, just to further investigate our anti-metastatic hypothesis. We've added some important biomarker studies in this design which weren't in the other design. And I think I want to go back to, whereas in traditional drug development, the safety aspect is almost a given. You know, when you're modulating gene expression like we are, and especially if you get into human trials, the delivery system and the payload, having any kind of safety badge like we've had is a very important validation of the whole preclinical R&D engine, multiple shot at goal, and the business plan. Okay, so we have a rare and world-class genetic toolkit, one of a handful of companies in the world. I hope that I've convinced you that we have at least a very simple business plan and a drug development model which can give investors the type of protection I think they need rather than the casino style of investment. We have net cash of 22 million. And I hope that I've backed up the claim that we're a world leader in genetic medicine. Thank you.